What is the structure of a promoter sequence? The promoters recognized by sigma 70 consist of two 6-nucleotide sequences called the -35 box and the -10 box. They are named such because they are located 35 and 10 nucleotides before the transcription start point. Transcription start point is also called the +1 nucleotide, and it is the first RNA nucleotide incorporated into the mRNA molecule. It is often an A, adenine, and is usually flanked by a C and a T. Understanding how scientists determine what the sigma 70 promoter sequence is will help you better understand the structure of promoters and how to identify them in a given DNA sequence. Calvin Harley and Robert Reynolds analyzed the known transcription start points of 263 housekeeping genes in the E. coli genome and genes in the plasmids and phages of E. coli. They determined how conserved each base of the -10 and -35 sequences are. By conserved, we mean how consistently a given nucleotide is found in a particular nucleotide position of the promoter. For example, in the promoters of the 263 housekeeping genes, a T is found in the first position of the -35 box 78% of the time, meaning it is highly conserved. Let's now look at the whole consensus sequences for the -35 box. There is a T in position 1, 78% of the time, a T in position 2, 82% of the time, a G in position 3, 68% of the time, an A in position 4, 58% of the time, a C in position 5, 52% of the time, and an A in position 6, 54% of the time. The consensus sequences for the -10 box is a T in position 1, 82% of the time, an A in position 2, 89% of the time, a T in position 3, 52% of the time, an A in position 4, 59% of the time, an A in position 5, 49% of the time, and a T in position 6, 89% of the time. Harley and Reynolds also learned that the space between the -35 and -10 box is conserved and is 16 to 18 nucleotides in length, with a 17 nucleotide spacer being most common or most conserved. This space between the -10 and -35 boxes is dictated by the amount of space between the two DNA binding domains of the sigma subunit. The DNA binding domains of the sigma subunit specifically recognize and bind, like a lock and key, to the -35 and -10 sequences of the promoter. If the -10 and the -35 boxes were 19 bases, or 14 bases apart, the DNA binding domains of sigma would not line up with the -35 and -10 sequences, and transcription would not initiate. What happens when the sequences of the -35 and -10 box stray from the consensus sequence? Promoter sequences with some differences in sequence will still be bound by sigma 70, but not as efficiently. This means less mRNA will be expressed from the promoter. These are called weak promoters. In contrast, a strong promoter has a sequence that is very close to the consensus sequence, and transcription initiation is efficient, resulting in higher amounts of mRNA. Not all phage and bacterial genes require high expression and a strong promoter. Can you think of a phage or bacterial gene that requires low gene expression and may have a weak promoter?